Okay. So any doubt from what we have done in the last class?
Sir, just one thing that the graph for the rate of photosynthesis, it's only for chlorophyll A, right?
Rate of photosynthesis? No. So it is of the process. No? This graph which you are seeing here, no? This graph, the action spectra. This graph is for the process. It is not for a particular pigment or anything, right? It is for the process, and that process is photosynthesis. So here what we have actually did is that, see, we have used this 400 to 700 wavelength, right? And here what we did that, here in this, let's say we started seeing, let's say first we have passed the wavelength of 400 and see what is the rate of photosynthesis, then 410, 420, 440, 460, and in this way we have seen till 700, that what is the rate of photosynthesis. And what we find ultimately that at this 2 point, the rate of photosynthesis is maximum. So now from here we don't conclude that it is chlorophyll A or chlorophyll B or it is xanthophyll or any other pigment, carotenoid, right? When we overlap these two, now next step is when we overlap. Now this is the action spectra, so the question can come that which pigment is contributing the most, right? Which pigment is contributing the most? So for that, what we did here, we overlapped one by one the action spectra with the absorption spectra. And what we realized? We realized that the action spectra of photosynthesis and the absorption spectra of chlorophyll A, they almost match, right? They are very much identical to each other. Is this point clear? They are very much identical to each other. And this gives us the conclusion that chlorophyll A is the major photosynthetic pigment. Is it clear? From just by this graph, you cannot say this is of chlorophyll A or chlorophyll B. This is for a process called photosynthesis, right? And this action spectra matches maximum with chlorophyll A, and that is why we call it as the major photosynthetic pigment. Is this point clear?
Yes, sir.
Now, here one important line is there. Let's read this line. So just read this line, this one which is given in italics. Read this line. Can you say that? Read it. Read. Can you say that?
Can you say that there is a complete 1-to-1 overlap between the absorption spectrum of chlorophyll A and the action spectrum of photosynthesis?
So can we say that?
Yes, sir.
Can we say?
Yes,
okay.
sir.
So let me show you the graph. Now here you see, is there a complete overlap? See here. Is there a complete overlap?
No, not complete, but not complete, but they are similar.
The peaks are matching, right? The peaks are almost matching, but there is not a complete overlap. See, this graph is like this, and this graph is something else. So there is no complete overlap. Understood? So here, if I ask you the answer of this question, then here what you will say? No, there is no complete overlap. Then the question comes, why no complete overlap? Why no complete overlap?
Because
because
when the chlorophyll A graph almost decreases to 0 rate of like 0 on the scale, but the rate of photosynthesis does not reach the 0 mark, and it does not stop.
Yes, this is the observation. But if I ask you why chlorophyll does not stop, even if the absorption of chlorophyll A is minimum, as we can see here in this graph, at this point you can see the absorption of chlorophyll A is minimum or 0 almost, but still there is some amount of photosynthesis here, some amount of photosynthesis here. Why is it so? The reason is other photosynthetic pigment, right? Because there are other photosynthetic pigment. They also have a role. Other photosynthetic pigment like chlorophyll B, carotenoid, they also play some important role, right? And these are called accessory pigment, right? Accessory pigment. So it is just like that. Let's say you are going to watch a movie, right? And in that movie there will be a leading actor. Let's say any movie in which Salman Khan is the actor. So Salman Khan is the main actor. But it's not that the whole movie only one actor will be there. There will be many other actors, although they are not that important. Every time when you say who is the actor, you will say Salman Khan. But other actors are also having some role in the story. So here the other actors are the accessory pigment. And who is the main actor? Chlorophyll A, right? So overall the movie not just need the main actor, it also requires other actors. But the main actor in the movie is Salman Khan. So this is something like, this is the identical scenario. So that's why we say that chlorophyll A is the major photosynthetic pigment, but not the only photosynthetic pigment. Can we say that?
Yes, sir.
Yes. So clear this point. So this
Yes.
is a very important question. So keep this in mind. Now let's move ahead. So this graph you understood well. Any doubt in this graph?
No, sir.
So there is an absorption spectra shown here. This is the absorption spectra. This is the action spectra, right? And this is the overlapping of absorption and action spectra. So let's say if I give you one graph like this, right? And here I plot a graph like this. Then one graph like this, right? I label this as A and I label this as B. And then if I ask you, what does A represent? What does B represent? What you will say?
A represents the rate of photosynthesis and B represent the absorption spectrum of chlorophyll A.
So if you are calling this as absorption spectra, so A you should call as the action spectra, right? Action spectra of photosynthesis. Understood? So in this way, similarly here also they can give you this graph and put instead of chlorophyll B, there will be A, B, and C, and then you have to find out what is what. So I think this you can easily now do. Clear this point?
Yes.
All
So
clear?
one more question. Can the rate of photosynthesis or the action spectra ever reach to 0?
Can the rate of photosynthesis? Yes, of course it can reach to 0 if it is beyond the visible range. See, there is a radiation called PAR, photosynthetically active radiation, and how much it is? 400 to 700. Right? Photosynthesis will take place only in this region. Beyond this region, the action spectra will be 0. No photosynthesis. Right? In between this, one or the other pigment will keep on absorbing light. So somewhat there will be contribution in the photosynthesis. But beyond this, the action spectra will be 0.
Okay.
Clear?
Yes. Yes, sir.
So let's move ahead. Now, this have we talked about reaction center, accessory pigment, light harvesting complex in the last class?
No, sir. No.
So here, if we before proceeding, let's see here one important point. Can you recall Engelmann's experiment?
Yes, sir.
What was the experiment? Conclusion of the experiment. What was the conclusion?
Like the red and the blue spectra absorbs the more amount of light. In red and blue spectra, the chlorophyll absorbs the more amount of light.
Yes. So basically, it says that photosynthesis is maximum in blue and red, right? And then at that time, we say that he was the first to study the action spectra of photosynthesis, right? So now we actually have studied the action spectra again at different wavelength. So can we say that these graphs, now read this line, these graphs together show that most of the photosynthesis take place in blue and red region of the spectrum. Okay? And then next, here it is not all, it is most of the photosynthesis. And next line, some photosynthesis does take place at other wavelength of visible spectra. Other wavelength of visible spectra. What I said, just the question which you asked, that in between 400 to 700, there will be some amount of photosynthesis keep on happening, right? But it has to be within the visible range, the visible spectra between 400 to 700. Beyond that, the photosynthesis will be 0, right?
Yes, sir.
Now, chlorophyll is the major photosynthetic pigment responsible for trapping light, right?
Yes.
So here chlorophyll means it is telling chlorophyll A. Understood? Okay. Other thylakoid pigments like chlorophyll B. Now, here they are only telling chlorophyll is the major photosynthetic pigment, but it is not including chlorophyll B because they are mentioning chlorophyll B in the accessory pigment. Understood?
Yes.
So if the question is asked, chlorophyll B is the major pigment responsible for trapping light, is it true? False, right? Chlorophyll B is considered as the accessory pigment. So clearly given here, chlorophyll B, xanthophyll, and carotenoid are accessory pigment. Right? What they do, they will absorb the light and transfer it to the chlorophyll A. Later on, just we will see that chlorophyll A will be called as the reaction center. But here, last line, and this is very important. Here, 2 points are given, right? It is the function of accessory pigment. If question is asked, what is the function of accessory pigment? What is the function of accessory pigment? So first you will say that, first function what you will say, that it helps in wider absorption, wider range of absorption, right? One can absorb, one pigment can absorb only at a fixed wavelength, But if there are many pigments, so they will absorb at different wavelength. So the wider range of absorption will take place. That is point number one. Second benefit is that this chlorophyll A prevents the this accessory pigment protects the chlorophyll A from photooxidative damage. Understood? So second, this accessory pigment protects
Okay.
chlorophyll A from, here it is given, photooxidation. So we will call it photooxidative damage.
Okay.
Understood? Now,
Yes,
what is
sir.
photooxidative damage? See, chlorophyll A is a molecule, right? It is a molecule, although earlier we used to study it, now it is out of syllabus, that there is a head, there is a tail like the structure, means that it is a large molecule, right? It is a large molecule. Now, if light fall on this molecule and if the intensity is very high, then this pigment may get damaged. And this damaging of pigment is called photooxidation. So it protects it from photooxidative damage. Is this point clear?
Yes.
So two function of accessory pigment is very much clear. Now let's move ahead. Now, earlier we already have seen that the photosynthesis is has two steps. One is called light reaction, another is called dark reaction. So now we will talk about light reaction. So for light reaction, you will say, sir, light is required, isn't it? Light is required for light reaction. Now we will see. Now we will see how these pigments are arranged. So we know that there is a chlorophyll A, which is the major photosynthetic pigment. So I'm just highlighting it here. This pigment over here, as you can see, I'm shading it with black. So here, can you see? This is called as the reaction center, right? So how many? So what is reaction center? So here I will write chlorophyll A. Chlorophyll A is acting as the reaction center. How many molecule of chlorophyll A will act as a reaction center? One molecule of chlorophyll A will act as the reaction center. Clear? So definition of reaction center is clear?
Yes.
Right. Then this reaction center or chlorophyll A is surrounded by large number of pigment molecules, and these pigments are called accessory pigments. They are called
accessory pigments.
Accessory pigments. Now these accessory pigments we can also call them as antenna. Right? We will call accessory pigments or antenna. So we are seeing that reaction center, which is chlorophyll A, is surrounded by the accessory pigment or antenna pigment.
Yes,
right?
Sir.
Or simply antenna. Now,
sir,
this is yes.
Yes. Sir, you said like chlorophyll A, one molecule. So like one, how like one acts as a reaction center?
One molecule of chlorophyll A is present at the center, and that one molecule acts as the reaction center. And this one molecule is surrounded by large number of molecules of accessory pigment. Understood?
Yes.
And this accessory pigment can be.
So one per cell, one chlorophyll molecule per cell, we should consider.
Not per cell, no, not per cell. See, we are going to study the unit of light absorption, right?
Yes.
Absorption. So unit of light absorption is called as photosystem. We are coming to that, but since you asked, I will go in the reverse direction. Photosystem. Now, photosystem is made up of pigments, right? Photosystem has pigments, and these pigments will have reaction center, right?
Yes.
These pigments will have reaction center, and in this case, it is one molecule of chlorophyll A, and along with the reaction center or reaction center is surrounded by the accessory pigment.
Yes, sir.
Right? We will also call these accessory pigments as the antenna. Right?
Yes.
Antenna.
Yes.
These accessory pigments are also called as light harvesting system. Light harvesting system. Right? These are called light harvesting system. So now overall what you understood? This photosystem consists of pigments, right? These pigments include reaction center, and these pigments include accessory pigment. Accessory pigment is also called as the light harvesting system. Now, overall, this together, can we call it as photosystem? Just I said?
Yes. Yes.
Photosystem. This photosystem is also called light harvesting complex. Light harvesting complex. Now, here these are LHC. Now, first let me explain it once again, and then if you have again doubt, you can ask. See, The question which you asked, that now the point is that there will be several LHC. Unit of light absorption cannot be one. This is one unit of absorption. Several such units will be present. Means several such photosystem will be present. Several such light harvesting complex will be present, and all of them will be contributing to absorb light. Clear?
Yes.
Now one. So here now we are only studying about one LHC. It's not that one cell will have one LHC. One cell can have several LHC present in chloroplast, and specifically where membrane of thylakoid. The membrane of thylakoid can have several LHC, light harvesting complex. Now our focus is to just study one LHC. So one LHC. So LHC is also called as photosystem, isn't it? LHC is also called as photosystem. Now this photosystem is having, means the photosystem is made up of pigments. As mentioned here, photosystem is made up of pigments, right? It is made up of pigments. Now these pigments include the reaction center, that is chlorophyll A, and it includes the accessory pigments. Now one common confusion students do is that they will read here light harvesting system, and they will think that light harvesting system and light harvesting complex both are the same thing, right? So they both are not the same. Remember, see, here it is given. I'm reading it, so underline it. The light harvesting complex are made up of hundreds of pigments, molecule bound to protein. Now, bound to protein means there are a large number of pigments. So how these pigments are together? So together they are with the help of protein. So this point is clear that LHC will have 100 of proteins. Now, what does these pigments are? So each photosystem has all the pigments except one molecule of chlorophyll A, forming a light harvesting system called antenna, right? So each photosystem has light harvesting complex, and that light harvesting system, and that light harvesting system do not have chlorophyll A. Means if you add chlorophyll A in the light harvesting system, it becomes light harvesting complex or photosystem. Is it clear?
Yes, sir.
Is this point clear? So just once again, I will mention it here in a very short form, right? So what I will say, LHC means light harvesting complex. So light harvesting complex is light harvesting system. LHS, light harvesting system plus accessory or plus reaction center. Plus reaction center, that is chlorophyll. Understood? Now this light harvesting complex is also called as photosystem, right? And this light harvesting system is also called as accessory pigment. Is it clear? Accessory pigment. Now this photosystem is of two types: photosystem 1, photosystem 2, right? Photosystem is of two types: photosystem 1 and photosystem 2. So both are photosystem. So both will have reaction center made up of chlorophyll A. Here the chlorophyll A will absorb at PS1. It absorb at 700 nanometer, and here the chlorophyll will absorb at 680 nanometer. So that's why this is called P700 and this one is called P680. Understood this point or not?
Yes. Yes, sir.
Right? So this point is 100% clear. See, from here, the concept of what is light harvesting complex, what is reaction center, what is accessory pigment, what is antenna, what is the relation between light harvesting complex, light harvesting system, all are very important. What is the difference between photosystem 1 and photosystem 2? That is also very important. So one difference, as I said, that the absorption, right? One absorb at 700 nanometer, another absorb at 680 nanometer. Now, this one and 2, this 1 and 2 numbering is not done on the basis of their discovery, not on the basis of their presence during the electron transport. This 1 and 2, 1 is discovered first, 2 is discovered second. That's why 1 and 2. It is on the basis of their order of discovery, right? It is given clearly that these are named in the sequence of their discovery and not in the sequence in which they are functioned during the light reaction. Understood this point? So this is also very important one. Sometimes you may get a statement, right? That the PS1 and PS2 are named so as per the sequence of their as per the sequence in which they function during light reaction. So that is false, right? That is false.
Yes.
They are named on the basis of sequence of their discovery, not the sequence of their function.
Light.
Clear?
Yes, sir.
So again, I am reading one statement. Tell me whether it is right or wrong. So the thing is, PS1 and PS2 are named so on the basis of sequence of their function, not on the basis of sequence of their discovery. True or false?
False.
False. Right? Very good.
Yes.
Correct. Now another statement. So another statement is light harvesting complex include reaction center.
True.
True. Light harvesting system include reaction center.
False.
Photosystem is the unit of absorption of light in the thylakoid membrane.
Yes, sir.
Yes, correct.
True.
So I think this point is clear. Any
Yes.
doubt in this overall?
No, sir. No, sir.
No doubt. Okay. So now let's move ahead. So here, because we are talking about the light reaction, right? We already
Yes.
know that. We already know that the photosynthesis is divided into two steps. First is the light reaction, another is the dark reaction. So when we talk about light reaction, then light reaction include a very important process which is called photophosphorylation. Photophosphorylation, right? Photophosphorylation. Photo means light, and phosphorylation means formation of ATP from ADP. So we can say photophosphorylation is the light reaction in which ATP is formed with the help of light. Now, this ATP formation or this photophosphorylation basically is divided into two types. One is called cyclic photophosphorylation and another is called non-cyclic. Right? Cyclic and non-cyclic photophosphorylation. Both are part of light reaction because photophosphorylation itself is a part of light reaction. Then, now first of all, we will see the non-cyclic photophosphorylation. So let's see this non-cyclic photophosphorylation. So here, can you see this graph clearly? So now here if you see non-cyclic photophosphorylation. So here you can clearly see LHC is mentioned, right? Light harvesting complex. LHC is the light harvesting complex. Now here LHC means photosystem also, right? Now which photosystem is this? This is photosystem 2, which is a type of LHC. Then here LHC is given to which photosystem it is? Photosystem 1. So the first point that, so it is about the non-cyclic photophosphorylation. So the first point about non-cyclic is it requires both PS2 and PS1. It requires both PS2 and PS1. This point is clear. Now, if you see here, light, because both are photosystem, so both require light to function, right?
Yes, sir.
Both will require light to function. Both will require light to function. So here also light is there. You can see here also light is falling. Now here which wavelength of light will be there at this point? PS2.
Sir, PS2 680.
680. Very good. 680 nanometer. And at PS1?
700.
700 nanometer. Very good. Right. So now when the light of 680 nanometer fall on the light harvesting complex or photosystem 2, the reaction center present here, it will excite electron. Right, and this excited electron will go to the electron acceptor. It will go to electron acceptor. Now the electron acceptor is PO5T, not given in NCERT and not that important as per NEET also. But yes, the electron acceptor is PO5T. If you forget it, it's fine, no problem. But just remember there is an electron acceptor, right? So electron goes to the electron acceptor called PO5T. Now this movement, can we call it as the uphill transport or uphill movement, right? So this excitation is uphill. Then these electron from the electron acceptor goes to PS1. From here it goes to PS1. But in this process, there is a step called electron transport system. Right? Can you see that? Electron transport system. Now this electron transport system, with the help of electron transport system, you can see ATP is formed from ADP, right?
Yes.
ATP is formed from ADP. Then the electron finally reach to LHC, means the photosystem 1. And here also the light 700 nanometer will excite the electron and electron will go to the electron acceptor. And from here, it will go to NADP plus. NADP plus and it forms NADPH. Is it clear? So the electron which excites from PS2 is finally accepted by NADP plus and the final product form is NADP.
NADPH.
Is the electron returning back or not?
No, sir.
No, that's why it is called non-cyclic.
Non-cyclic.
Right?
Yes.
Fine. So one by one, let's now characterize it. If we see here at photosystem 2, there is a process which we call as the splitting of water. Here you can see water is split into 2H plus, 2 electron and 1 oxygen. These two electron enter inside the photosystem 2, and this acts as the source of electron, permanent source of electron. So if I ask you the continuous source of electron during non-cyclic photophosphorylation is
Water.
what you will say? It is water or splitting of water, right? Splitting of water. Then if I say the final electron acceptor, who is the final electron acceptor?
Photosystem 1.
It is the photosystem 1. Or
Yes.
after photosystem 1, electron is not accepted. See here
No,
only you see electron
NADP.
accepted. NADP
H.
H.
Yes.
Final electron acceptor is NADP plus, and it becomes NADPH. So now here, first one by one, both photosystem 2 and 1 is involved here. Second character is splitting of water. Second character is
Splitting of water.
splitting of water. Now splitting of water take place at PS2, not
2.
PS1, right? Then splitting of water will produce
2 electrons.
It will produce 2 electrons, right? And it is acting as a source of electron, but it also produces oxygen. So oxygen is produced, right? Now H2O act as the source of electron, isn't it?
Yes, sir.
Finally, can we say that who is the final electron acceptor?
It's
Final
NADP plus.
Right? The final electron acceptor is NADP plus. Then further, what we can say here? What further can we say? Here, this conversion of NADP plus into NADPH require an enzyme called NADPH reductase. NADPH reductase. Is this point clear? And of course,
yes,
here ATP is produced through ETS. So these are the characteristics of non-cyclic photophosphorylation. Is this point clear? Now,
yes, sir.
Here, few additional points are there which we will see, right? And the additional points now I will write. This here you can see there is uphill transport, right? Now I will use different color. So let's say here this is the uphill transport, as you can see here, uphill. Now can we call this as downhill?
Yes.
Downhill is movement from electron acceptor to photosystem 1. Now can we again say this as uphill? Uphill,
yes.
Movement here, again uphill, yes. And this movement again downhill. Can we say yes. that? So yes. Electron is moving first from LHC to electron acceptor, uphill, right? Then electron acceptor to photosystem 1, downhill. Then photosystem 1 to electron acceptor, again uphill. And then then the electron acceptor to NADP plus, again downhill.
Downhill.
So uphill, downhill, uphill, downhill. Right? Now they may ask you only a particular step. For example, if I say that electron move from photosystem 1 to the electron acceptor, this is uphill or downhill?
1 to electron, it's uphill.
Uphill. Movement from the primary electron acceptor to PS1 through ETS, it is
downhill.
Downhill. Clear? So the uphill downhill concept is also very clear. Now, this non-cyclic is also called Z scheme. It is also called Z scheme. Now, Z scheme is because of its characteristic shape. It appears like a Z shape. See here. It appears like a Z shape. So it is called Z scheme. And this is formed because of redox potential scale. See, one thing again, you keep it in mind. I will explain with example. Let's say electron is moving from A to B. Electron is moving from A to B. Tell me, which one has higher redox potential? If electron is moving from A to B, so which has higher electron potential? Not
B.
Electron potential, sorry, higher redox potential.
B.
Higher redox potential. Tell me.
B. Sir, A.
See, remember, whenever an electron moves from A to B, so B will have higher redox potential and A will have lower redox potential, right? Electron always moves from lower electron potential to higher electron potential. So here if you see, the movement of electron is on the basis of the redox potential, right? If I'm sometimes telling electron potential, so that's a mistake. Just consider it, I'm telling redox potential, right? So higher, from lower redox potential to higher redox potential. Is it clear?
Yes.
So why is it called Z scheme?
Because it's in the shape of Z.
Shape. How is this shape formed?
Due to uphill and downhill movement of electron.
See, here, this, read the last line. Last line important. Due to its characteristic shape, this shape is formed when all the carriers are placed in a sequence on a redox potential scale. Right? How is this Z scheme formed? When all the carriers are arranged on the basis of their redox potential scale, because electron move from lower redox potential to higher redox potential. Is this point clear?
Yes, sir.
So now let's move on ahead. So splitting of water, we have already discussed it, that one molecule of water will split. See here, 2 hydrogen is there, so it will split and form 2H plus. Now 2H plus, so there will be 2 electron also, and 1 oxygen is formed, right? So in order to form one molecule of oxygen, how many H2O you need? In order to form one molecule of oxygen, how many H2O is required?
2.
2 H2O, and this will produce how many electron?
4. 4
so
electrons.
as it is mentioned here, as you can see, 2 H2O break down into 4 hydrogen, 1 oxygen molecule, and 4 electron, right? Now, where does this splitting of H2O take place? So you will say at the inner side of the thylakoid membrane and near the photosystem 2. It takes place near the photosystem 2. See here, it is clearly given the water splitting complex. See, underlining it. Water splitting complex is associated with photosystem 2. If there is no photosystem 2, there will be no splitting of water. And where is it located? It is located onto the inner side of the membrane of thylakoid. Right?
Yes, sir.
Is this point clear?
Yes, sir.
Now, let's move ahead. So next is the cyclic photophosphor. Although phosphorylation we have already discussed that it is cyclic and non-cyclic, so we will straight move to the cyclic photophosphorylation. So here, as you can see, the cyclic one. Now, if you see here, what you observe, this P700 is what? Photosystem 1 or 2?
1.
Photosystem 1. Where is photosystem 2?
Not present.
Right. So when we say cyclic photophosphorylation, so here in cyclic photophosphorylation, what we observe, that the first thing is there is no photosystem 2. Only what present? Only photosystem 1 present. First difference. Second difference, if there is no photosystem 2, then will there be splitting of water? Just we have seen that splitting of water is associated with
photosystem 2.
Photosystem 2. So no photosystem 2. Rest of the point you can conclude by your own. No photosystem 2, no splitting of water. If there is no splitting of water means
no electron.
No electron and no oxygen. Electron can come from anywhere. It's not that electron is only, but here no splitting of water means no
oxygen
produced here. Now about electron also, let's discuss. If we see the photosystem 2 excites the electron.
Yes, sir.
Photosystem 2 excites the electron due to light. Which wavelength of light is here? Not, it is 700 nanometer. 700. It excites the electron goes to electron acceptor, and then through electron transport system it come back again to photosystem 1. So can we say that electron return back?
Yes.
Electron return back to photosystem 1. So who is the final electron acceptor?
Photosystem 1.
Yes. So source is also photosystem 1 and final electron acceptor is also
photosystem 1.
Photosystem 1. Now, if you see that whenever photosystem 1 is exciting the electron, it is returning back, right? It is returning back. So do it require a permanent source of electron? Does it require a permanent source of electron?
No.
no source of electron required, right? So
Yes.
even if there is no splitting of water, which is not producing any electron, still the process will continue. Is it clear?
Yes, sir.
Because H2O is acting as a source of electron, so here no source of electron is required. Also,
Yes.
because final electron acceptor is PS1, so no
Electron loss.
no NADPH is formed,
Form.
right? So no
Yes, sir.
oxygen, no NADPH. What is produced? But ATP is produced. ETS is taking place. So what is the product of cyclic photophosphorylation? Only ATP. What is the product of non-cyclic photophosphorylation? ATP, NADPH, and oxygen.
Oxygen.
3 products are there, out of which ATP and NADPH will be used in the dark reaction, and oxygen is a waste for plants which is released outside the plant.
Yes.
Understood? This
Yes.
point you understood well?
Yes.
So now it's your homework that you have to list down all the difference between photosystem 1 and photosystem 2, each and every possible difference. One more point I will like to add here. It is related to photosystem 1 and photosystem 2. So again, see it very carefully, right? See, if you recall the structure of chloroplast, so chloroplast, inside the chloroplast, there is stack of thylakoid like this. And this stack of thylakoid is called?
Grana.
What is it?
Grana.
Grana. And these, let's say 2 grana are there. So these 2 grana are connected to each other, isn't it?
Yes.
They are connected to each other.
Yes, sir.
Yes, Arya? Arya, can you hear me?
Yes, sir.
So here, what I just drawn is, one is the grana, right?
Yes.
This part is the grana, and this one is the stroma laminae, which connects to each other, right? Now, the purpose of drawing this diagram again is to explain you the location of photosystem, right? See, photosystem, we say that it is present in the membrane, but membrane can be, so stroma lamellae also have membrane, and grana also have membrane, right? Grana also have membrane, and stroma lamellae also have membrane. So now remember this point. When I say photosystem 1, so photosystem 1 is present in both stroma lamellae. Sorry. Photosystem 1 is present in
Stroma lamellae.
So here also the photosystem 1 will be present, and in the grana membrane also photosystem 1 is present. So photosystem 1 is present in both stroma lamellae and the grana. Where is photosystem 2 present?
Stroma.
There is photosystem 2 present. So photosystem 2 is present in the, it is only present in the grana. It is only present in the grana. Right? So here I am drawing photosystem 2. Photosystem 2 is not present in the stroma lamellae. It is not present in the stroma lamellae. Now, one thing also, if you notice here, where I have drawn the photosystem 2, inner part of the grana, right? Whereas the photosystem 1 will be present on the membrane, surface part, right? The exposed part. All are membrane only. But see, the difference you are understanding, this is the exposed part. And the red dots I have drawn for photosystem 2, they are the non-exposed part, right? So, which is called, although it is not again given in NCERT, but it is a good point, right? So just keep it in mind. So here, this photosystem 2, we call it, it is present in appressed region. Appressed region of grana. Which region of grana? Appressed region of grana. And PS1 is present in the non-appressed region. Non-appressed region. This point, first of all, tell me whether this is clear to you or not. The location of photosystem 1 and photosystem 2. It is clear?
Yes.
100% clear? Okay.
Yes.
So again, I'll ask question, tell me the answer, then I'll understand that you it is clear to you. So tell me, stroma lamellae will have which photosystem?
Photosystem 1.
1. Will stroma lamellae have photosystem 2?
No.
Grana will have which photosystem?
Both the photosystem 1 and 2.
Yes. So the non-appressed part or non-appressed region will have?
Photosystem 1.
And the appressed region will have?
Photosystem 2.
2. Understood this point? Now, on this basis, if I ask you, so here just think for a few seconds and then answer, right? Now, if I
Yes.
ask you, where will cyclic photophosphorylation take place? My question is very clear. Where will cyclic photophosphorylation take place?
Stroma lamellae and the non-appressed region.
Yes. So simply you can say grana, right? Grana.
Yes.
So if I say cyclic photophosphorylation, so cyclic photophosphorylation will take place in the stroma lamellae also,
Stroma lamellae.
stroma lamellae, and it will also take place in the grana. Now, if I ask you where non-cyclic will take place, non-cyclic
Appressed region.
photophosphorylation.
Appressed region.
See, when you are telling, so it will require both PS1 and PS2, no?
Yes.
Non-cyclic photophosphorylation need both PS2 and PS1. So now you don't say appressed or non-appressed. You simply say grana because it will require both appressed and non-appressed region. So
Yes.
non-cyclic photophosphorylation take place in grana only, not in stroma lamellae. But cyclic can take place in grana also and in the stroma lamellae also. Understood?
Yes.
Right? This point is 100% clear. From here, question can be asked. So keep this in mind. Now,
yes, sir.
On this basis, if you read this line, so it is underlined over here. See, the possible location where this could be happening is in the stroma lamellae, right? While the membrane of lamellae of grana has both photosystem 1 and 2, the stroma lamellae lacks photosystem 2 as well as NADP reductase. Understood?
Yes, sir.
The stroma lamellae not only lack photosystem 2, but it also lack NADP reductase enzyme, whereas the granule membrane have both photosystem 2 as well as the NADP
reductase enzyme.
This point is very much clear.
Yes, sir.
Right? So
yes.
This conclude this point. Now, next point again, and we'll quickly read through it. The excited electron does not pass on to NADP plus, but is cycled back to PS1 complex through the electron transport system. This cyclic flow result only in the synthesis of ATP. And not of NADPH. Right? Now,
yes.
Cyclic photophosphorylation, when it will occur? Only if the light beyond 680 nanometer is available. If light of 680 nanometer is present, then will cyclic take place?
No, sir.
No, right? Because then non-cyclic will take place. Now, if I ask you which is more important, cyclic or non-cyclic?
Non-cyclic.
Non-cyclic is more important because it gives us ATP, it gives us NADPH, it gives us oxygen. Without non-cyclic, can dark reaction take place?
No.
No, right? Because for dark reaction, both ATP and NADPH is required. Is it clear?
Yes.
So, this cyclic photophosphorylation only produce ATP, right? So when it will take place? If the light of wavelength 680 is not available, means beyond 680, means greater than 680, that is, if the light intensity of what intensity? 700 nanometer, then only the cyclic will take place. Is this point clear?
Yes.
So if I say that if light of 680 nanometer is present, will cyclic photophosphorylation take place?
No, sir.
No. If light of 680 nanometer and 700 both is present, then will cyclic take place?
Yes, sir.
No, still no. Because cyclic will take place only if 700 nanometer wavelength of light is present. If 680 is present, then who will be favored?
Non-cyclic.
So remember this point very clearly. If 680 is present, then even if 700 is present, who will be favored?
Non-cyclic.
Non-cyclic. If 680 is absent and only 700 is present, then who will be favored?
Cyclic.
Cyclic. So this is what you have to keep it in mind. So a lot of important points is covered here. So I will leave you here so that you can revise everything once again. So all the notes are available here. I have kept it saved. So you can go back, read everything, revise everything. And if still you have any doubt, you can ask in the next class.
Yes,
so today,
sir.
if you have any doubt, you can ask.
No, sir. No doubt, sir.
Okay, so I will stop the class today.